Ma'am,
Hello.
good afternoon, ma'am.
Good morning.
Oh, good morning to you. All
Will you
right, so we have ma'am to show us.
will you be showing the video?
You can show the video if you like, ma'am.
Okay, I could try, but it's not very good on the internet. It might be better for you to show it.
All right, ma'am, that's absolutely not a problem.
Yes. Why don't you show it, and then I will be here to answer questions.
Absolutely, ma'am. Just give me a minute.
Hello out there. This is Suzanne Anker coming to you from the SVA Bio Art Laboratory in New York City. I hope everyone is doing well, and I'm going to lead you through some of our visuals regarding our laboratory. It's a very special laboratory since it essentially is a cross between a 19th century and 20th century laboratory. We have live animals such as coral and fish, even some crabs and other kinds of sea creatures. We also have plants and taxidermied animals as well. You can see here that we are very much interested in tissue culturing. The pieces on the bottom under the grow lights are African violets, which have been made through the tissue culturing procedure. In our aquariums, we have been able to actually hatch skates, baby skates, which it is said that they cannot be hatched in captivity, but we have done this numerous times. On the bottom middle slide, you can see the skate embryo in the skate case. This is our area for molecular biology where we actually test for DNA as well as creating chemical gardens as well as paint with bacteria. Here are some students in the class who are working away on their projects. You can see on the bottom right that there is a class that is involved in cactus graphing. We have many collections. These are some that students can borrow to draw from. These are insects in resin or skeleton specimens or even the kind of ectoskeleton from a deceased coral polyp. Some of the other things that we have done, as you see in the top image on the left, is to use this clear and staining technique in which we take animals that have been deceased, we retrieve them from pet stores, and we do this kind of procedure that just allows the bones and cartilage to be stained. The piece on the right is another tissue culturing experiment in which we were able to tissue culture Venus flytraps, which we feed with flightless flies. And on the bottom middle image, we see a Madagascar cockroach that was given to us by one of our students as part of a video project, and we have managed to maintain their population. We started with 5, and now we have many, many more, using a kind of therapy by petting their backs with a paintbrush. Of course, everyone knows SCOBY, the symbiotic culture of bacteria and yeast. The piece on the left is a sculpture that has been made out of this material that is reminiscent of a carnivorous plant. We also see SCOBY in various colors, in which we have used hydrogen peroxide to bleach the material. Of course, mycelium and mushrooms is another one of our projects in the way in which we bring the idea of sustainability into the lab. The piece on the right is a gravestone called Agua that is made out of this material, and as visitors come into the exhibition, they're asked to spray the gravestone with water so that the mushrooms can bloom. This piece is by Andrew Siraki, who was able to work with samples of holy water from Catholic churches in Manhattan and Brooklyn. And what he found was...
Just give me a minute. There has been a technical issue. I'll just resume the video in a minute.
has created a number of very interesting pieces. This one on the left, called Hourglass, is a way to show the correspondence between chlorophyll and hemoglobin. And what she did here was to bleach out the chlorophyll of leaves, and when one puts an ultraviolet light on them, one sees that the chlorophyll can be red. Another piece by Tara on the right-hand side is called Blood, Sweat, and Tears, in which she collected her own tears and sweat, and using artificial blood, used a molecular technique to create these different pellets. Victor created a agriculture sculpture in which the kind of cycle of life is replicated through the debris from the fish as fertilizer for the plant. So it is a closed system. Here is a work of mine called Astro Eternal Return from 2015. This piece has circled the globe in many parts of the world. And here, this is an example of it in an exhibition called The Value of Food at the Cathedral of St. John the Divine in New York City. And when people came in to see this piece, they didn't believe it was real. They had never seen anything like this. So this is kind of an ode to the way in which agriculture is being brought indoors. And here you can see that if you shine a light on any of these plants, that they are still green, even though they appear to be fuchsia. We were also involved in several competitions in which we won medals for them. The first one was called Soiled, and it was part of a project for iGEM. And what we did is we collected soil samples from all over the 5 boroughs in New York City. It's very common that one sees an urban garden in one of the boroughs in New York because land is very scarce. We did a wide range of pH tests that would make the samples ready for the spectrophotometer. And you can see here that the colors are very much like a palette that an artist uses in their work. Here is the protocol for nitrate, nitrite, phosphorus, and other pH test procedures. We developed a polished copper board in chemical baths to be used as a relief mold for casting our microfluidic chip. And here you can see that we changed the hydrophobicity of the chip via oxygen plasma treatment. This is a maquette for the LED installation of materials. This is just part of a visual art project. And here you can see our diagrams and sketches for the soiled exhibition. This is our exhibition site at iGEM in Boston, where we have photos and our model, as well as the explanation for this project. What we were able to do with these soil samples was to create these visual maps of what the different kinds of nitrates, ammonia, sulfates, iron, etc., were present in each one of these samples. And as you can see here, the soil samples show us the relative amounts of each of the elements. The piece is very reminiscent of Damien Hirst's dot paintings, but this is actually data. Lo and behold, we won the gold medal for the Best Art and Design project in 2015. We were also nominated for the Best Integrated Human Practices, the Best Education and Public Engagement, and the Best Presentation. Another project coming out of our lab was called Myo Tomato, in which we attempted to insert myoglobin, which is essentially a protein similar to hemoglobin, into a tomato. And the idea here was to eliminate the animal from the ecological kind of situation that we have due to methane gas. What we did is we began the Myo tomato in culture in vitro, and sort of shows us that the history of the tomato, which starts in Central and South America, is really very reminiscent of an Aztec calendar. So visually, that was a very striking resemblance to us. Here are the steps in which we actually editing of DNA. We inserted the DNA into the tomato via agrobacteria, exposed the plant tissue to bacteria. We cultured the tissue. We induced and grew and rooted baby plants. And we grew the transgenic crop in our greenhouse or makeshift greenhouse in our laboratory. And then we analyzed it for protein content. And here you can see the gel electrophoresis indicating the myoglobin gene has been integrated into the tomato genome. We also made some mock products to go along with our presentation, such as diced Myo tomatoes, tomato soup, tomato sauce that are reminiscent of Andy Warhol.
Hi there, welcome to the SVA Bio Art Lab, where we've been designing the future of food for over 30 years. Good. This laboratory, the first of its kind, is where biologists and artists work together tackling some of the world's biggest biological concerns. Over the years, we've spawned some unique results, but nothing as successful, impactful, and delicious as this. Meet the Myo Tomato, the amazing protein-packed fruit that started it all. A biological breakthrough that changed the way we think about protein. I have something amazing to show you. Randy, get the thing.
As you wish.
Before the Myo Tomato, humans relied heavily on animals for their daily source of protein. With a growing global population and a strained agricultural system, we recognized animal protein was not an ecologically sustainable source. There had to be an alternative method. We put together some of the brightest scientific and artistic minds we could find, and the result was impressive. Take a look.
This will only hurt a little bit, JK.
In 2016, the BioArt team at the School of Visual Arts engineered the world's first fruit to produce the same protein, myoglobin, found in animals, the Myo Tomato. They found plants to be deficient in megadoses of protein necessary for a nutritional human diet. So they translated a DNA sequence found in beef and inserted it into a tomato's genome. The result? One little tomato possessed the same amount of protein found in an 8-ounce steak. This breakthrough creation caused quite the stir. It seems not everyone was a fan of this amazing scientific achievement.
GMOs are dumb and stupid and poison.
Of course we're not happy.
However, in the year 2030, mad cow disease almost completely wiped out the cattle industry, creating an urgent need for the Myo Tomato's amazing protein power. And soon, hearts, minds, and mouths were open. The Myo Tomato ushered in a global movement that ultimately ended our need for animal consumption. Not bad for a group of art students, if you ask me.
Thank you for listening, and I'm ready to take any questions if you have any. Well, there is one question here about whether we can make other watery fruits and vegetables more nutritious, and I'm sure that is correct. Because we are not scientists at the Bio Art Lab at the School of Visual Arts. We really do not have the expertise to go much further. We did go a little bit further and found out that the myoglobin actually entered into the tomato's genome, but we couldn't tell how much of the protein was there or where it was. It would take at least a longer study, perhaps even a PhD, to develop this. I may add that this was done in 2016, well before the Impossible Burger or the other burgers that came on the market that were made from different vegetable proteins, that from the Big Whopper, etc. I'm not sure whether in India that was an item. So in many ways, this is a speculative project, but with the right funding and expertise, I'm sure that this could actually exist. There, are other kinds of projects we thought about doing, such as duck l'orange, which is a very famous French dish where you could insert the DNA of a duck into an orange, and maybe it would taste like duck l'orange, or certainly fish and chips, in which you can insert the DNA of a flounder into a potato. So these are just some of the different kinds of thoughts that we have. And as artists, we think very metaphorically. And the lab is a bit different, as you have seen, than your traditional scientific lab, because it is really one in which the projects we produce have to be related to not only science, but to art as well. So it's really being involved with two different discourses. And all of the work that you've seen here were all students or former students, and everyone was an artist, except actually we had some scientific help as well. And any questions, I'd be happy to take from you. Another question is, even after the tomato being genetically modified, would it be considered as an option for vegan eating? Well, this is a very good question. I don't have the answer to that. You would have to speak to a strict vegan in that sense. Another question here, where do you see BioArt's role in the future? I think that BioArt is a great companion to scientific investigation because the thinking processes are quite different. We've done a number of things that said couldn't be done, such as hatching baby skates in our aquarium. We've also been able to change the color of agar. We've made black agar, for example, in which if you put the phosphorescent bacteria on, you can see it much more clearly. We've used molecular cuisine techniques for other liquid forms, and right now we are studying issues about plastic as well as sustainability. Yes, Myo Tomato is not actually available, but if anyone has any way to market this, I would certainly like that very, very much. um, Another comment here, this was a visual treat, love the creativity. And I think that's really important to demystify science, because this course is not a science course. It's an art course that uses science to make art. And I think that that's really important to engage younger people. I know that many artists have been afraid of science, particularly the kinds of precision that is required in scientific investigation. But I think as a team, artists and scientists can create spectacular kinds of work, both scientifically and aesthetically, and really advance both of the practices in other ways. I mean, you saw that in our soil data exhibition, the relationship between Damien Hirst's work and the data of the different nutrients in soil. There are many crossovers here that can be presented. And I know that in many scientific journals now, there are ways in which Photoshop and other software programs are eliminating artifacts from research, which is a problem, because is the best image the best science? This becomes a question. And okay, here, I think an actual product that's available in the market that's similar is the Arctic apple. It's genetically modified and doesn't turn brown when exposed to air. They have a huge regulatory timeline, but it's been finally approved in the US. Well, thank you for that information, because I did not know that. There's also the question here, we turn to obviously seeds and seed banks, and how seeds might be perceived in situ or ex situ. And what I mean by that is a seed in a seed bank, in keeping the either Arctic apple or the Myo tomato or whatever in storage, may not behave the same way as if it would in the wild. So we have a lot to learn about that. My piece, Astroculture Eternal Return, was also based on NASA's project of growing vegetables in outer space. And what the astronauts found initially, these were the Japanese astronauts, was that when plants grow in anti-gravitational situations, they sprout two sprouts rather than one. So there's much to be learned about our botanical others under different conditions. And of course, the idea of plants being the necessary companions to humans in order to sustain life is of great interest to me. If anyone is coming to New York at any time within the next couple of years, essentially, please let me know and come visit us in the Bio Lab. I'm very sorry I could not come to India, which I have never been to. But I think that the issue of bioart, biodesign, the intersection of these subspecialties has become a kind of international foray. There have been many exhibitions and conferences, et cetera, around the world in this way. In fact, the piece that I showed you, the astroculture piece, was shown in Korea at the Daejeon Biennial, in which the entire museum was given over to bioart. And what's also interesting about seeds is you can't take them with you. That essentially I had to go and get Korean seeds and Korean plants, because otherwise you need a passport for your plants and seeds. Another question: during the making of Myo Tomato, did you face any difficulties, and how did you troubleshoot? The only difficulties we faced was really going further with it, because we were not equipped at that point to further investigate its possibilities. Anybody else here?
Ma'am, I have a question. Could you open to it?
Sure. Can you speak a little louder?
Can you hear me now?
Yes, I can.
So you spoke so much about art and biology interacting with each other, creating these spectacular products. How exactly does one work in this field, given you require so much innovation, so much thought process, so much out of the box ideas that are usually impossible to achieve? How do you
How do you do that? You bring
do that?
together a lot of creative people who really think up ideas that probably would not be funded by the National Endowment of the Sciences, and just sort of continue to brainstorm about this. You know, we were fortunate enough that we have an art building with all the facilities that we need, whether it was rapid prototype machines or chemical baths, etc. But the kind of expertise that the scientists have, and we invite scientists to come to our lab to help us understand some of these processes. For example, when we tested different cookies and vegetables for GMOs. It's kind of the reversal, where inviting an artist into a laboratory is one thing, but inviting a scientist into an art laboratory is the reverse. And I think that works much better.
Have you, as a bio artist, ever faced the block that usually people go through? There's this phase where everything is so difficult, coming up with ideas, getting the funding, commercializing them, implementing it.
Yes, well, I've been very lucky. I am chair of the department, and I have my own fine arts building of 54,000 square feet. I'm sorry, I don't know centimeters. And my idea was to connect the handmade with the technological through the arts. And when I decided that we should have all of these kinds of computer output machines, it made perfect sense to have a bio art lab, since biology is technology. And I went to the president of the school, who I meet with regularly, and I told him that I would like to fabricate a bio art lab. And he said, "What's that?" Okay. It's a very hard concept to get your mind around, but he allowed me to do it. And because we are a private institution, I don't have to fundraise. I can just use the departmental money in the ways in which I find to be most valuable. So it's a very, very lucky situation like this. The other thing is, in 2004, I wrote a book with the late sociologist of science, Dorothy Nelkin, called The Molecular Gaze, Art in the Genetic Age. It was the first platform about art and genetics and social policies. So this is a triangular kind of endeavor, because sociologists or philosophers don't ask the same kind of questions that scientists or artists ask. And I think one needs to consider all of those aspects. And the book was published by an august scientific press, the Cold Spring Harbor Laboratory Press, which is where Barbara McLaughlin and McKibben, excuse me, and also Mr. Watson was, among others. So it can be done here. It's just a question of really having enough information and dedication to get your idea across.
All right, ma'am. Thank you so much. Thank you.
Well, thank you for inviting me. And Gunter is here from Vienna. That was, hello, Gunter. And I'm sure I have other friends in here somewhere. And feel free to email me anytime you want to. And essentially, if I can help in any way, I'd be happy to do that. Okay?
I'd love to take you up on that.
Yes. And hopefully one day I'll see you in India. Okay?
One day, hopefully.
Yes. Okay. Well, good morning, everyone. It's only 20 to 6 a.m. here, so I'm up very early. Okay. Okay. Bye-bye.
All right. So for the next session, before the next session, we have a poll which